Welcome back, everyone, and thank you for returning. We are ready to start the final session. So it's my pleasure to introduce Patricia Raciti, a pathologist at Paige, who is going to talk to us on integrating digital diagnostics into clinical practice to maximize patient impact. So Patricia, thank you. Over to you. Thank you very much. I think you're on mute. Or...
Thank you, Dr. Sneed. Can everyone see my slides and hear me okay?
That's perfect. Thank you.
Wonderful. So thank you for the kind introduction, and to everyone at Pathologist and Innovate UK for the invitation to speak at this visionary conference. Today I'll be sharing my thoughts with you on a topic I'm very passionate about, integrating digital diagnostics into clinical practice to maximize patient impact. First, a bit about myself. After training at Memorial and Columbia, specializing in hemepath and dermpath, I became a general practice community pathologist as well as a pathologist at Paige, where I do receive a salary and am an equity holder. I sign out everything. What are my areas of expertise and interest? Well, in the development of AI products, their testing and validation, studying the benefits and drawbacks of these products, and advising on the presentation of AI data for optimization of efficiency and safety. So when evaluating AI products, pathologists often ask, do I have an effective algorithm? Does the AI work? Often, the starting point for evaluating the performance, clinical utility, and efficacy of such systems is in silico validation and standalone performance. How does the AI perform against ground truth, looking at sensitivity, specificity, and so forth? This is often at the slide level and can be on curated data sets, generating figures that might look familiar to you, ROC curves, some sort of performance of the AI product compared to a pathologist. There might even be static outputs on the whole slide image in the form of annotations or heat maps. These may tell a more nuanced story about performance, where perfect slide level accuracy might translate into so-so heat map accuracy. But all too often, these starting points are not imbued with a complex understanding of pathology and can actually be just an ending point. And in fact, there are many, many more questions that typically come up. Many of them are fundamental and foundational, and others are very practical. Is this system safe? Can I understand why it fails? Will it save more money than it costs? All of these are very important. Why? Because these products will always be evaluated in the hands of a human and should be evaluated as such. Unless we plan on replacing pathologists, which we don't, standalone performance cannot be the end game. So to determine whether your AI works, whether the patient will ultimately see a benefit, is about understanding how it works in the hands of a pathologist, which heavily depends on human factors, satisfaction, learning curve and proficiency, decision-making impact, and design and workflow factors, usability, when the AI is presented, and the way it's presented. Is it interpretable? Is it evaluable? And notice the word trust. It's a topic I've been thinking a lot about. What does it mean? And do we actually want it? I'll talk more about that later. And this belief is shared by other thought leaders. There's recent and clear momentum in the clinical research field to focus on the human-AI combination, as outlined in these timely letters and studies. And this at Paige is what we've done. We conducted a study where we supported human decisions were the endpoint in assessing clinical utility. So I'd like to delve into our clinical work on prostate cancer detection, evaluated in the hands of a pathologist. We focused on prostate needle core biopsies where significant time-consuming and diagnostic challenges can exist, though they vary. In the top core, detection is not a challenge, as this tissue has a heavy cancer burden, and instead grading and quantification become the challenge. In the bottom, if there is cancer, it's not obvious. So finding the cancer needle in this haystack of tissue, so to speak, is the burden the pathologist bears. There are tools that the pathologist has access to in the first instance. Levels and consensus opinions really are the most effective in this largely interpretive task. But in the second instance, a task of pure visual detection, blinded universal re-review of negative cores is really the only current way to address the challenge. But realistically, this is time-consuming, potentially costly, and just as error-prone. So we ask, can we develop a product to enhance and speed up prostate cancer detection by pathologists in needle core biopsies? The study involved 3 AP board-certified pathologists, all with fellowship training, but none in GU. They worked in community hospitals, and they had a very high comfort level using web-based software. The product we tested, Paige Prostate Alpha, was developed using a multiple instance learning approach, free of pixel-wise annotations, that was pioneered earlier by Campanella et al. The study consisted of 2 phases, each lasting 2 days, and separated by a 4-week washout period. Day 1 of both phases consisted of an overview of the slide viewer, instructions of the study, and a live demonstration. So we walked participants through how to analyze and view a whole slide image in the viewer. The second day in both phases is what varied. In phase 1 of the study, day 2 consisted of an 8-hour period where participants reviewed a set of digitized H&E stained prostate needle core biopsy slides. And they were tasked with classifying the slides as suspicious for cancer or benign. This was called the unassisted phase. In the second day of phase 2 of the study, participants reviewed the same set of digitized slides, this time after Paige Prostate marked the region considered statistically most likely to harbor cancer on images deemed to have cancer. Again, pathologists were tasked with classifying slides as suspicious for cancer or benign, having been provided this information by Paige. This was the Paige-assisted review in phase 2. This is an example of what participants would see when Paige Prostate marked the slide as cancerous. You could see a black cross over the region of highest suspicion. And let's take a look at the data set. 232 slides were reviewed with the ground truth of the diagnosis of cancer or benign having been established from the corresponding pathology reports. Looking at these plots from left to right, 40% of these whole slide images were in fact cancerous and 60% were benign. Each grade group was represented, but not all Gleason score combinations. And the majority of cancer cases comprised Gleason grade group 1. About 1/3 of the cases harbored cancers that were smaller, less than 1 millimeter in length. So in the study, we sought to answer these questions. How does pathologist sensitivity change with Paige? Are they better able to recognize smaller tumors with Paige and lower grade tumors with Paige? Are they faster at classifying slides with Paige? And importantly, how do they interact with and incorporate Paige into their evaluation? With Paige, pathologists, we found, more accurately detected cancer. You can see sensitivity plotted on the y-axis and the performance of each pathologist with and without Paige, along with their average on the x-axis. And across the board, sensitivity of detection increases for each pathologist in phase 2 when they use Paige. On average, this was a statistically significant improvement in sensitivity from 74 to 90% with the use of Paige Prostate. What about smaller, lower grade tumors, the hardest and most time-consuming to detect? Again, looking at pathologist sensitivity with and without the use of Paige Prostate by cancer size and grade, we see with Paige, pathologist sensitivity in detecting cancers of all sizes increased, but the increase was especially pronounced for small cancers. And with Paige, pathologist sensitivity in detecting lower grade cancers increased. And from an efficiency standpoint, we looked at the average time it took to classify a whole slide image. With Paige, pathologists classified all slides, benign or cancerous, faster. This time reduction was statistically significant overall for cancerous slides and for cancerous slides with less than 1 millimeter of cancer. Now let's take a deeper look at the pathologist AI interaction with a few select examples of whole slide images, specifically looking at how the product potentially influenced or did not influence the participants. All of these next 3 slides show cancer in prostate cores by ground truth, and let's see how the pathologist and Paige classify them. In the tables, X means an incorrect classification. So in these cases, incorrectly calling a slide benign, and check means a correct classification. In this case, correctly calling the slide cancerous. Here, we see that all 3 pathologists incorrectly called the slide benign without Paige. And then, despite Paige's correct classification in phase 2, only 1 pathologist changed their mind to cancerous when shown the Paige results. The rest maintained a benign classification. Another slide, this one of treated cancer, especially tricky. Without Paige, 2 pathologists correctly classified the slide as cancerous and 1 pathologist incorrectly classified the slide as benign. Here, Paige incorrectly classified the slide as benign, And with the Paige output in phase 2, only 1 pathologist changed their diagnosis to benign and 1 of them switched to cancer, and 1 kept their original correct diagnosis. Finally, this disturbing but very real case of a small focus suspicious for cancer under a tissue fold. Here, all the pathologists in both phases and Paige incorrectly classified the slide as benign. So what are the key takeaways from looking at these data on select whole slide images? Pathologists changed their opinions. Pathologists and the AI can be wrong or right. Sometimes pathologists agree with the AI, and sometimes they don't. And pathologists and the AI are most often right and produce better results together. It's just the next level of questioning that we at Paige are thinking about, and the field, I believe, as a whole needs to delve further into. And that is, how do we get the AI to be right more often and in more types of cases? And how do we get the pathologists to trust the AI when it's right and correct it when it's wrong? And we need to take a step back and ask ourselves, what are we really trying to achieve versus what is the end goal? If the end goal is to have the pathologist perform better with the product than the pathologist alone, or the product alone, then our analysis is done. But what are we really trying to achieve? Aren't we really aiming for pathologists to perform better with the product because of the product? If that's the case, we need to dig deeper into these performance improvements and ask ourselves, how much of this improvement can be attributed directly to the product versus chance re-improvement based on reanalysis or seeing a previously overlooked area? And for that, we really need to take a critical eye at these corrected interpretations, as you see in this table, where the pathologists were originally incorrect in their assessment, but then corrected their assessment on the same slide after being shown the results of an AI. Specifically, we need to look at the percentage of the corrected whole slide image in which the AI presented the correct result to the pathologist. Ideally, that's 100%. We also need to look at the percentage of the corrected whole slide images in which the AI presented the incorrect result, and yet the pathologist still got the answer correct. So we did this exact analysis in the study I presented. In phase 1 of the study, without Paige, the aggregate number of whole slide images classified incorrectly, false negative or false positive, by pathologists was 87. In phase 2, with Paige assistance, 61 of those 87 whole slide images that were initially incorrectly classified became correctly classified. They were corrected. Why the improvement in phase 2? Because of chance or because of Paige? Let's take a look at the Paige classification on those 61 slides, and therefore what was shown to the pathologist in phase 2 by Paige. Turns out, of the 61 slides that pathologists corrected in phase 2, Paige correctly detected cancer in 59 of them, most of them, missing only 2. This suggests that Paige, more than chance, played a role in helping a pathologist get to a correct diagnosis. So in summary, for this study, we showed that there were statistically significant gains in average sensitivity without statistically significant changes in specificity, important from an efficiency standpoint. Pathologists were able to recognize smaller more well-differentiated cancers and able to classify slides faster. And there was significant insight into the pathologist-AI interaction. So I want to focus on this human-AI interaction piece and question, how do we optimize it? Is it about trust? I'd observe that no product is perfect. And if a pathologist always trusts the product, the pathologist's performance characteristics might simply converge with those of the product. And the goal is to have the pathologist perform better with the product because of the product than the pathologist alone or the product alone. And so I'd advocate that what we really want is algorithmic vigilance, not blind trust. So how do we get to this state of algorithmic vigilance, where the pathologist knows when to correct the product? I'd argue providing the pathologist with as much information about the tool as possible. The more the user knows about the product's strengths and blind spots, the better vigilance pathologists can have when using the product. And at Paige, we strongly practice this and believe in it. This deep understanding of the product begins at the algorithm design stage with deep involvement with the pathology team. And it continues in the product validation stage with continued deep involvement with the pathology team. Why is pathology involvement so crucial? Only a pathologist will know and think of these aspects of evaluation, thinking of mimickers, variants, and the spectrum of disease. As a pathologist, I want to know the algorithm's performance on mimics of cancer, on mimics of benign. I want to know how it performs on poorly defined and in-between categories like ASAP, how it performs on variants of cancer, as well as its performance by grade and size, which we've done. I'd also want to know how it performs on floaters and artifacts, performance on a tissue floaters or tissue that wasn't detected. How does it perform on folds, out of focus area, and other scanning and preparation artifacts as we saw in that example? How it might show unknown bias, such as performance based on patient characteristics like gender, age, race, ethnicity, things a pathologist might never think of. And finally, how it performs in other encountered use cases potentially outside of the intended use, for instance, in prostate, small cell cancer, or intraductal cancer, non-acinar adenocarcinomas. And there's much more to assess that involves close work with pathology and AI teams, UX teams, and even scanning vendors to assess all of these other facets. Interpretive, as we think of Gleason scoring, quantitative, as we think of measurements, computational, how long does it take to visualize the results of a tool? UX are the results presented in an understandable way, a way that I can't possibly misconstrue them, and in a way that I can evaluate them quickly and that they don't take more time than me giving the diagnosis de novo. And finally, the pre-analytic variables, as I touched on. How does the slide quality and the scan of the slide impact how the algorithm will produce results? Let's go through one more example. This is a benign prostate core showing seminal vesicle, a known mimic of cancer. Potentially not so much on the image on the left, where it's clear that it's probably seminal vesicle, but in the image on the right, it's not inconceivable that this might be misclassified as cancer if this were the only focus, especially, and if potentially other cores showed 3+3 cancer that were small. So we showed this slide to 16 pathologists, 2 of which were GU specialists. You can see pathologist 5 and pathologist 14 in black. And this time we asked them to assess the slide as benign, suspicious for cancer, or to defer their classification until immunohistochemical stains or levels or a colleague's opinion might be available. In all, 3 pathologists deferred classification. 6 called the slide cancerous, and 7 called the slide benign. Paige was run on the slide. How did Paige classify it? Paige correctly classifies the slide as a true negative, benign. Now, pathologists were presented with the same slide again, this time with Paige prostate's results, and tasked with the same question. Was it benign, cancer, or did they defer? Well, let's look at the changes in their assessment. 3 of those initial deferrals corrected to true negatives after being presented with Paige results. 2 of those initially false positives corrected to true negatives after being presented with Paige results. And 3 of those initial false positives converted to deferrals after being presented with Paige results. So in summary, false positive evaluations with Paige decreased to 1, and true negative evaluations with Paige increased to 12. So shifts in the right direction. But total deferrals, though, remained unchanged. And while in this case, deferrals were an important step in the right direction, considering these pathologists had previously misclassified the slide, the goal of deep algorithmic understanding and vigilance of the pathologist is to convert more of these classifications to correct ones. And so in conclusion, I'd argue that integrating digital diagnostics into clinical practice to maximize patient impact is about optimizing the human-AI interaction. This is achieved by a pathologist-driven AI design and evaluation so that the end pathologist user can be armed with information about the product's strengths and weaknesses for that tissue and cancer type. This puts the pathologist in the best position to perform better with the product because of the product than either the pathologist or the product could do alone. So I'd like to add that Paige prostate will be studied prospectively in a pathologist-led study as part of the NHSX Phase 4 award, something we're very proud of. And the study will measure real-world health economics to drive system-wide adoption across the NHS. This is led by the University of Oxford under Dr. Claire Verrill, who's incredible. And at Paige, I'd like to conclude that we're committed to creating and designing and understanding how products work better with the pathologist, not simply generating standalone data. This is our driving philosophy, and this should be the driving philosophy of the field overall. So I'd like to thank the audience and the organizers. I know I've ended a bit early, but please do feel free to contact me if you have any questions. But otherwise, I very much look forward to your questions later today. Thank you so much.